everybody. It's great to be here. It's always fun being part of the Global Genes and this rare disease community, which is very open to collaboration. It's something which I really love about this field, and which is very different from the rest of most other scientific fields or disease areas. So it's really a pleasure being part of this. I mean, it's going to be great kicking this off with some of the research tools which are available to you. So a little bit on my background very, very quickly. So I am a scientist by training. I have a PhD in experimental medicine with a focus in molecular genetics. And I took a non-traditional route, going the biotech and entrepreneurial route and founding Modelis back in 2018. So to kick this off, we'll begin discussing some of these cell-based models, or these in vitro models, which are available for a variety of research uses. And we'll use this time to go through some of these models quickly and go a bit deeper into them later on in the discussion. So among these cell-based models, the first ones that we'll touch on are cell lines. So these can be obtained from human or animal tissues, such as fibroblasts that are usually obtained through a skin punch. Then we have iPSCs that are stem cells that can be differentiated or turned into basically any cell or tissue type. And these can be generated from skin or blood cells and are patient-specific. But in the absence of any patient-specific lines, we do have these CRISPR-edited cell lines which are available. And these are using CRISPR or genetic engineering techniques to mimic very specific patient mutations or very specific genetic variants in the cells where you don't have any patient-specific cell lines available. Thank you. So while the models on the previous slide are what we call two-dimensional models, there have been recent advancements in the generation of what we call organoids, which are these tiny three-dimensional structures that can be grown using iPSCs, or the induced pluripotent stem cells, to study specific tissues or organs in a disease state. And these can be made into brains, livers, pancreas, even muscle cells. And so while in vitro means in the glass and typically refers to cells in a dish, in vivo here means in living and refers to simply animal studies, or more generally studies in live multicellular organisms. This is really a topic which I'm very passionate about since I use these very regularly. We at Modelis use these for drug discovery. And so while mice are typically the most common, most well-known of these animals, there are many others that can be used to answer a wide variety of research questions, from the very small model organisms, such as fish, zebrafish, frogs, or xenopus, flies like Drosophila, and worms, C. elegans, and it goes all the way to the larger mammals like pigs, dogs, and other non-human primates. And each of these organisms have their own strengths and limitations, and while each can be used to answer specific questions, having multiple ones in your tool belt can really help you answer the widest range of research problems. And as for the types of therapies that are typically available to you, the most traditional ones are these, or we'll quickly go over these traditional categories, traditional modalities, which are small molecules. So think of your typical pills like Advil, which often can be taken orally. Then we have biologics, or protein-based therapies, which are usually engineered full-length proteins such as antibodies or enzyme replacement therapies, all the way to peptides that are short proteins like insulin, which is probably the most common of these, or the most well-known of these. And while we did name these traditional therapies, it's not because they're out of date, it's just because they have a very long and historical use as therapeutic classes. And finally, one more approach, which we'll just quickly touch on, one more tool in your tool belt which can be used, which is leveraging computational approaches to identify or repurpose drugs. And so aside from all the biological, so the in vitro and the in vivo approaches, there's another aspect which we just quickly wanted to touch on, which are these computational-based approaches, also known as in silico tools. And these are most often AI-driven, that look to predict molecules by leveraging data such as molecular targets to design very effective or very specific drugs to very specific target proteins. And that can be incorporated or aligned with any of the other strategies we had discussed before, any of the other tools we had discussed before. And also kind of going back to an important point, each of these can help you answer specific questions. And one of these is usually not the be all end all. And the broadest range of tools in your toolkit can really help you answer the widest range of questions.